Thank you very much for the opportunity to present at this venue. My talk will be focused on deuterium magnetic resonance imaging for cancer and immunotherapy. I'm a physician scientist at the Roswell Park Comprehensive Cancer Center, and my research spans multiple departments. My primary appointment is in the Department of Pediatric Oncology. So that's, as a physician, I'm actually a pediatric oncologist who treats children with cancer with a focus on bone marrow transplantation. My scientific interests are in immunology, and as you will see, in radiology as well. So my translational research program is focused on many projects, but I will cover two of them today. All of them have to do with deuterium, and I will explain that in a second, but I wanted you to see where my research is really focused, and that's on bench-to-bedside translational research. So I'm at this interface of preclinical into early clinical development, and that's where my passion really lies, taking things from my own laboratory into early phase clinical trials. The first thing I'll talk about is using vivo systemic deuterated water labeling to facilitate imaging of cancers. And then the second portion of my talk will focus on how we can use that in cellular immunotherapy. So what is deuterium? I mentioned that word in the title. So deuterium sounds scary, but it's actually a stable, so that means non-radioactive, heavy isotope of hydrogen. So hydrogen is the first element in the periodic table. It has a weight of 1. Deuterium is special. It has a molecular weight of 2. So it has an extra neutron. So that's why it actually does not result in a change to becoming a radioactive isotope. But what's interesting about deuterium is that it looks different to the MRI machine. An MRI machine is a magnetic resonance imaging device, similar to NMR, those of you who are in chemistry, but it's used to image the body using the magnetic waves. So why am I interested in using deuterium for MRI? So it turns out that in vivo deuterated water labeling, so if you were to drink deuterated water to get it through the IV, it results into deuterium incorporation into building blocks of proliferating cells. So my first research project on deuterium was actually in my postdoc training, which is where I was using deuterated water labeling to look at DNA synthesis in T cells. So how does that work? So your body actually uses water to make DNA. And if you look at the other things that it serves as a substrate for, it's basically every pathway that a dividing cell needs. So it's used for protein synthesis, cholesterol synthesis, lipid synthesis, and nucleic acid synthesis. So at first, I was using it as a method to quantitatively detect newly dividing T cells by extracting their DNA from target organs and immune organs involved in graft versus host disease. But what I will say is on the detection end was a very expensive and complicated detection method, gas chromatography mass spectroscopy. So I had to partner up with an analytical chemist who could do the actual extraction for me as well as the quantification. Then we had this idea of using deuterium labeling in imaging. So what we ended up doing is at first we were studying T cell kinetics, and I had to extract T cells out of every possible tissue. It was a very painful process and took a lot of mice. But what we stumbled upon was that deuterium could be used in magnetic resonance imaging. So I thought, wherever I see my T cells, I could probably see deuterium. And therefore, I could jump from extracting cells out of tissues into whole body imaging to detect where T cells were abundant. So why that makes a difference is because really, as I hope I have already explained, deuterium incorporation is a proxy for cell proliferation. So we had this idea of maybe we can use this to image tissues with high proliferation rates non-invasively with whole body imaging by MRI. So the first papers we found describing the use of deuterium as a nucleus in MRI imaging was from 1980s. And there was really like 2 or 3 papers on this and then pretty quiet. Part of it's back then, deuterium was a little bit more expensive and not as readily available. And what it was used for in this experiment that I'm showing you here was simply as a contrast agent. So if, let's say, a rat or a mouse or a human drank deuterated water for weeks and weeks and weeks, tissues would light up, like the CSF that you're seeing in the rat brain on the right-hand side. And the rosette you're seeing on the left panel is labeled like 1, 2, 3. Those are reference solutions. They're cross sections through a vial containing a solution of 1% deuterated water and regular water, 2, 3, 5, etc. So the higher content of deuterated water and regular water resulted in more imaging signal. So this idea came to me when I was a postdoc at the NCI, and we filed a patent at that point. Initially, I was using this in graft versus host disease, but the idea was really to use it in proliferating cells and for imaging cancer. And the first paper to describe that we published in 2023, so fairly recent. What it involved was we provided deuterated water systemically to a mouse that had been tumored with pancreatic cancer cells from a human being. Actually, in this one, it was the one I'm showing you here on the slide is from colon cancer, human colon adenocarcinoma. So it's an immunodeficient mouse in which we've implanted a tumor, and you can see that in the depiction in panel C. And then we provided the drinking water to the mouse for 7 days while the tumor was growing. And as you can see in the red, so what are the three circles in the cross section? So the three circles are, the larger one is the tumor-bearing leg. The other bluish circle is the leg that does not get a tumor, so it's an internal control. And the little circle is a reference solution that contained 10% deuterated water in regular water. And I hope you can see through the different slices through the tumor is that the red signal indicates higher intensity, and that is the deuterium MRI picture now of the legs and the reference solution. And the tumor's full of deuterium. So what we were hoping to see was if you provide deuterated water to a mouse or a person, if they have cancer, the cancer cells will take up the deuterium and then will light up on MRI. Why is that useful, actually? Because sometimes you can see cancer. So for example, if you go back to the previous slide, the tumor's pretty big, right? If we're looking at the black and white picture, that's sort of the anatomical imaging. And you can see the one layer is bigger than the other. And if you were to look at the mouse, you could see the tumor pretty well. But in other settings, let's say in a human being in a home, you have a tumor internally in the abdomen or in the chest. You can't actually see it without doing some imaging. And sometimes when the tumor is quite small, it looks just like normal tissue. So you can't tell, is it a normal lymph node or is it one involved with lymphoma? So any kind of ability to distinguish a functionality of the tissue, like is it proliferating or is it quiescent, is very helpful in that setting. So after this was all published and the patent was filed, et cetera, I was recruited to Roswell Park, and I had to set this imaging up again now in a place that had never really done multinuclear imaging. So I mentioned MRI before. It's widely used, except that I would say 99% of MRI uses protons. So our body's full of the regular protons, the 1H now, not the 2H, not the deuterium. It's abundant. So you can stick a person inside a magnet into the special magnet, the MRI machine, and the different densities of protons, let's say in the lung compared to muscle, compared to other tissues, will show up. And so you can see lungs differently than brain, than abdomen, et cetera. Now for deuterium, you have to have a special setup. And that requires multinuclear capability. So I had to set this up both in my animal imaging, but also for clinical imaging, because at this point I was ready to write the clinical trial. So this will be a story now of what it actually takes to put this into practice. So now we're talking about translation between bench to bedside. And I had to set this up in parallel because I continue doing experiments in mice, as you will see, as I will describe on subsequent slides. And I'm also ready to translate some portion of this research into clinical practice now. So when I came to Roswell, we had a different intensity of a magnet. So magnetic field strength that are measured in Tesla. So typical clinical scanners are 3 Tesla. So the first set of experiments I was doing at the NIH were done at 14 Tesla and 11 Tesla, so high field, really high field, which gives you good sensitivity. But most institutions will not have that kind of MRI. Furthermore, in clinic, we use 3 Tesla. So everywhere in every kind of academic center, if you have an MRI, it's going to be probably a 3 Tesla or a 1.5, but 3 Tesla is very common. So what we needed to do was to really ensure that this method could be deployed in patients at 3 Tesla. And then I had to really test this in mice at 7 Tesla, which is sort of represents what we might be able to see at lower field strength deployed in people. And the difference between the 7 Tesla and 3 Tesla can be kind of okay if you're imaging a person, because much bigger body than, let's say, imaging a mouse at 17. So we established that we had clinical feasibility to image mice at 7 Tesla. And we also resurrected my mala that I had used at the NIH. So on the right-hand side, you're seeing imaging, proton images of a tumor in the leg of a mouse again. And on the left-hand side, you see the colored panel is showing you a deuterium overlay over that region. And what we were able to establish was that we could see that tumors very well with our imaging strategy, the labeling strategy we had previously used at 7 Tesla, which was really good because we went from 14 to 7 Tesla. But also what we wanted to make sure is how long was it really necessary to label with deuterium, with deuterated water? How long does somebody have to drink it for before you can see the tumor? And our first set of experiments, it had been 7 days. So that was sort of our standard thing. But it was also done at 5% whole body water concentration. Now in mice, that's very easy to achieve. It requires 1 IP bolus of 700 microliters of saline made in deuterated water. And then we maintain 5% continuous labeling in the drinking water in the mice. But to achieve that in a human being, the scale up is tremendous. The amount of water you would need to drink every day to actually get to, let's say, 5% would take a long time. So we wanted to figure out, can we do this with shorter labeling periods, but also labeling periods that did not require us going all the way to 5% body water. So we safely were able to do that in mice, and we also saw that we had enough contrast between tumor and sort of quiescent tissue. So that's really reassuring. This kind of set us up to figure out what it would cost us to do this in human beings, and how much time would be required for us to do this on a clinical trial. So right now we're looking at 3 consecutive days of somebody drinking 250 mLs of deuterated water at 70%, and then they would be able to go inside the MRI machine. Now that's really feasible because if you're talking a week, it could still be done. So for example, a patient comes into Roswell with newly diagnosed cancer, you have to do the staging scans for them. And sometimes the scheduling does take that long with MRIs, but beyond a week, you really don't want to be delaying any kind of decisions being made in terms of treatment and et cetera. So 3 days puts us at a really nice target. The expense of the deuterated water it's expensive, but the biggest expense is actually the scan time. So the MRI machine is a very complex piece of equipment, and the insurance charge for a scan for an hour is about $4,000. So the deuterated water costs about $1,700 per liter, and we wouldn't even need that much per patient. It's really the scan time. So now we knew, okay, we're actually in the ballpark. We're exactly where we need to be. So what we also found out by collaborating with other investigators at Roswell is that we could not only detect the initial diagnosis of tumor, but we could also detect treatment responses. And where this becomes important is pseudoprogression. So that's a new term I'm introducing now. What does pseudoprogression mean? So for example, a patient is diagnosed with cancer. You see it on their initial scan, and you start treating. A few weeks later, a few months later, you rescan them, and you want to see, did the tumor shrink? Well, it turns out in about 1/3 of the cancer patients, it looks like the tumor gets bigger, even if there's actually a response. So pseudoprogression is when you think the tumor has gotten larger, and therefore you're thinking it's progressing clinically, but in reality, it's actually responding. And how do you know that? Because if you scan them, let's say 6 months later, you'll actually see that finally the tumor will shrink. And initially we thought, well, how often does that happen? But I'm telling you, it happens pretty frequently. So 1 in 3 patients undergoing cancer therapy will experience something like this. And of course it matters, because what if they're on an investigational therapy and you don't know if they're going to respond, and you decide, you know what, they're going to need to come off the trial. And then they might not have another trial to sign on to. We've heard of several situations where patients were taken off a study drug, and they had a 1-month washout before they could begin another study, only to realize on a rescan as they were entering the other study and about to start a new drug that they had indeed responded to the previous one. So now you've lost some time. Let's say you've been off of an effective therapy for a month. So a lot of people are going into clinical trials not because they want to just be sort of our guinea pigs, is because they don't have any other treatment options or curative options. So we don't want to be taking patients off of effective therapies. So I've talked a lot, but really what I wanted to show you on this slide here is another model in which we had an ovarian patient-derived xenograft implanted into mice. We did sequential imaging before and following a highly effective antibody-dependent cell-mediated cytotoxicity treatment. So this is where a chemo that would otherwise be very toxic if given systemically, but when it's connected or chemically bound to an antibody that specifically allows its delivery to the tumor site and not anywhere else, it can be quite effective. And what I'm showing you in the bar graph is in the red is tumor size, and in blue is contrast, deuterium contrast. So I hope you can focus in on day 2 post-treatment. You see how the tumor has actually enlarged in size, but actually the deuterium signal has gone down. So that is the key time point in this model where we're seeing pseudoprogression. Well, you might say 2 days, does that really matter? Because, in 2 weeks' time, you'll actually see the tumor regress. But it matters because in patients, the pseudoprogression can last for weeks and months. So the time frame here is compressed, thankfully, because it's really expensive to image both mice and humans by MRI. But it gives us this idea that potentially we have a way of seeing pseudoprogression. And I'll remind you again, this is a very safe imaging technique. So MRI does not involve using ionizing radiation or X-rays like CAT scans or X-rays. And deuterium can be drunk in safely, especially at the doses that we're going to be using at 2% body water. So there's about 500 patients that have already received deuterated water, including children. And some of the labeling was done for kinetic studies, like the one I was embarking on as a postdoc for analytical chemistry, sort of end detection. But some of it was done in endocrinology studies, et cetera. So there's a body of literature to suggest that it's very safe to drink. It looks and tastes like water. So we're basically ready for prime time on this now. So where is deuterium MRI available worldwide? It's not many places. And most of them are not connected to a cancer center where they have a volume of patients to treat. So I highlighted in pink here where deuterated water has been used, and anything that's starred indicates that it's been used clinically. So we're hoping to actually, I need to update the slide because the clinical trial for using deuterium MRI has been recently opened on August 8th, just a month ago. So when I first came to Roswell, I'd already mentioned I had to set this up in parallel, both in the clinic and in the lab. So I pretty much talked about what it took to do this in the lab and showed you some data that we generated at Roswell. Well, what did it take to set this up at Roswell? So what I started is I had a 3 Tesla GE MRI system in the Department of Radiology. And the things that you're seeing now listed in white are all the things I had to put together to make this MRI machine be able to detect deuterium in patients. So I had to get a multinuclear hardware upgrade, multinuclear software, which was licensed to me through GE, a radiant filter from Cambridge, UK, or collaborators there, and then the head coil and a body coil. So if any of you are physicians, if you've ever ordered an MRI for a patient, you have to specify which body region. And even if when you're ordering a regular one, a proton MRI, you have to specify. Why is that? Because a special piece of equipment needs to be put in ahead of time. So if you're going to be imaging the head, you're going to need to put a head coil in. If you're going to image the body, you need to put the body coil in. So for deuterium, we now have the capabilities of imaging brain tumors as well as things in the abdomen. So this is my investigator-initiated trial. So I did my part pretty much more than a year ahead of the trial opening. It was already approved and ready to go. But what I was waiting on was equipment. So far, I've really talked to you about deuterated water. But deuterium can actually be put on other molecules. So it can be attached to glucose. And then you can actually look at glycolysis of tissues. Now, it's well known that tumors are glycolytic. That's the Warburg effect that was published in the 1940s. And a lot of our imaging techniques that are functional are actually based on that principle of higher sugar metabolism in cancer cells compared to regular cells. And the way we've exploited that for clinical imaging is using radioactive sugar. So if any of you have heard of PET scans, positron emission tomography, that's what that deploys. That uses radioactive sugar molecule, which then is administered to the patient either IV or orally. And it will that sugar will be preferentially taken up by the cancer cells. And that's just uptake. So it sort of gives you a picture of that. But for some diseases, for some tumors, it cannot be used. So for example, one place like that is the brain. Brain runs on sugar. So when you're reaching for that candy bar, it's not actually you. It's your brain that needs to have that sugar. And the brain tumor and the entire brain will have really high uptake of glucose. So it doesn't help you to use a PET scan in the brain. But with deuterated glucose or other isotopes of glucose, you can actually trace the downstream metabolites. So you can detect both the glucose, but also the lactate that's made. And that actually allows you to then distinguish a brain tumor from the rest of the brain, because the brain tumor presumably will make more lactate. So the first cohort that's open right now on my clinical trial is primary CNS lymphoma patients for whom we will be using this imaging. So for them, there's preclinical data to support and clinical data to support that patients who have more aggressive CNS lymphoma will have higher lactate levels in their CSF. And when imaging was done with carbon-13 labeled pyruvate, another technique, metabolic imaging technique, it also suggests that patients who have poor responses tend to have their tumors light up on that kind of scan. So cohort 2 will be used in graft versus host disease. That's sort of my flagship project, where it's the T cells that we're going to be imaging. So graft versus host disease, I will just allude to it, is a disease after bone marrow transplant. It's not something you can catch. It's iatrogenic. It's something that is a result of our therapy. And that's where the new immune system is given to the patient. But it unfortunately recognizes more than just their leukemia. It recognizes their normal organs. And this is largely T cell mediated. And T cells, when they activate, are highly glycolytic. So tissues that become infiltrated by activated T cells, like gut and liver, light up on this kind of scan. And that had shown in mice, and now we're hopefully going to be able to see that in human beings. And why that matters is because there's currently no established imaging for graft versus host. The diagnosis is based on biopsy, so that's pretty invasive. And if you're tracking responses over time to different therapies you're using, it's also going to be really difficult to track that and rebiopsy a patient who's just undergone a bone marrow transplant. So here I'm describing my next clinical translation plans, and that will be deuterated water in CNS lymphoma patients. Why do I need to open up another imaging trial in CNS lymphoma? That's because some patients are not going to be eligible for the deuterated glucose uptake. Some patients are diabetic, some patients are on GLP-1 inhibitors, and they should not be drinking a huge bolus of water of glucose. It's not huge, but it's 60 grams. It's not a minor dose. So for them, water perhaps would be a better approach. And also there's other tumors I'm always thinking about. So as I mentioned, I'm a pediatric oncologist. So I see young adult patients as well as children with cancer. And one of those cancers is Hodgkin's lymphoma. It's highly treatable now. So more than 90% of the patients are actually cured. And in these patients, we're primarily using FDG PET CT scans for staging and response assessment. So patients who do really well end up having 3 FDG PETs, initially a diagnosis, interim response assessment, and a conclusion of their therapy. So that's 24 years of background radiation that results just from the scans. Forget the chemo and other things that we're doing to them. And if they have progressive disease, which is still very treatable, they'll end up with 4 or more scans. So that, each FDG PET exposes you to about 8 years of background radiation. So if we can eliminate this from something that's very curable, then that would be ideal. So I'll switch gears, and this part of my talk is going to be quite short because this project is a little bit further out, a little bit downstream from, upstream rather, from the early clinical development. It's still very much currently being tested in vivo and mouse models. And that is ex vivo deuterated water labeling for visualization of immunotherapy trafficking. So most of you probably know about chimeric antigen receptor T-cell immunotherapy. It's something that's revolutionized the field of oncology, using the immune system to fight cancer. And this is done with taking autologous T-cells from a patient and engineering them to express the receptor against a cancer cell. So that's worked very well, actually, in leukemias, lymphomas, and myeloma, so blood-based cancers. There's now 6 approved drugs, which I've listed here. They're FDA approved. So this has revolutionized the care of patients with refractory hematologic malignancies. But the next frontier is actually solid tumors. So currently, the success of adaptive cell therapy in solid tumors is not really a success. It's kind of a disaster story, only 10% response rates. And why is that? What's different about blood cancers versus solid tumors is really Target specificity. So there's tumor antigen heterogeneity. So for example, on leukemia cells, if it's a B-cell leukemia, CD19 is universally expressed on the cancer cells. But in tumors that are solid tumors, there may be several different types of antigens that are expressed on various sections of the tumor. And the other really important aspect is they impair T cell trafficking to solid tumors. Now, when we do this in mice, and I mean CAR T engineering, we have slam dunks. And we definitely can get our CAR T cells in if they're expressing the right antigen. But in human beings, it's not always the case. So if we look at more physiological models, not like where you inject an immunodeficient mouse with a human tumor, but if you actually look at a model where you're putting, let's say, a pancreatic tumor into the pancreas, then it's a lot harder to get your cells in. And the reason for that is these tumors form a wall, basically, where it's really, really hard for cells, any cells, to really traffic in. And then once they get in, the other issue could be the immunosuppressive tumor microenvironment. So if the cells are able to get in, but they're not able to fire up and actually kill the tumor. And this is known because tumor TILs, so tumor infiltrating lymphocytes, have been detected previously for decades. It's been known that the immune system can enter the tumor, but whether it kills it or not is a whole other story. So currently, how do we use CAR T cell trafficking is something I'm going to focus on because that's what I'm developing deuterium imaging for. So we currently look at CAR T cell trafficking really by looking at flow cytometry and peripheral blood. Well, that's fine for leukemias and lymphomas, but that doesn't actually work for solid tumors, right? Because we don't know, did your cell therapy make it into where it needs to go? You can perform immunohistochemistry on tumor samples, but then you have to rebiopsy the patients. That's not ideal. There are currently imaging methods that exist too, but they rely on either radioactive tracers, so they expose patients to radiation or expose your cells that are hopefully delivering the cure to radiation, or an nanoparticle labeling, which involves mechanoporation, which can compromise cell membrane integrity and function in vivo. And then you can also introduce genetic markers, so they will allow your cells to light up in vivo if you administer a drug, for example. But there's potential to introduce immunogenicity. So there's guidance on this from the FDA. It's encouraging the development of non-invasive imaging methods for cell trafficking and monitoring. And again, FDA also started an initiative to reduce unnecessary radiation exposure for medical imaging. So we think deuterium has a lot of potential in this space. So I initially was labeling cells in vivo and looking at their DNA. So I knew that my T cells and graft versus host labeled up from systemic administration of deuterated water. But what if I did it in cell culture? So for example, if we make media in deuterated water, we can actually potentially track these cells in vivo after they're administered systemically. So the patient isn't drinking the deuterated water, but it's your cells as they're being expanded. Let's say your CAR T cells, as you're growing up and expanding them in culture, in the last day of culture, you can put them into special media and they'll pick up the deuterium. And then after the infusion, a few days after, you can perform the MRI and hopefully see them in the tumor. So we're a little bit far out from that at this moment, but we have a model of BiTE T cells, bispecific T cell engagers, so another form of cellular immunotherapy. And then this model, we've inoculated the mouse with a tumor, and then we ex vivo labeled our cells, and then we put in varying amounts, varying concentrations of T cells, varying numbers of labeled T cells. And for now, we injected them directly into tumor, and we've been able to see the deuterium signal go up after the treatment and then subside afterwards. So in the dotted line on the right-hand side, you can see pre-ACT, the deuterium signal. Day 1 after ACT has been administered is blue, and then red is day 2. So we're still working on this and testing this in different models, testing different concentrations. How much labeling do we need to give to, like, what percent media do we have to use, etc. But that's work that's ongoing in my lab. And again, I don't undertake anything in the lab that I don't plan to put into clinic. So future studies will involve more physiological models and hopefully patients. So at this time, I'd like to thank my team. I currently have 2 members of my lab as my technician, Joel Dusen, and then Chen He, who's my MD PhD postdoc who joined me a year ago. He's a radiologist by training, so it's very helpful to have a member of the team who knows how to read scans and analyze imaging data and sophisticated imaging data like deuterium. And then I will open this up for questions. You're welcome to contact me after the talk. One of the pictures I'm showing here on the very last slide is just how often different fields of science interact with each other. So if you look, for example, at basic life sciences and clinical medicine, that connection is really strong. But if you think about engineering, physics, which is MRI, how often do those connect with clinical medicine, or chemistry, for example? Those connections are not quite as strong. So I hope you appreciate that these projects that I described today are not only, moving from bench to bedside, but they're also connecting many different types of science. So from physics to computational biology to engineering to chemistry. And it really makes for a fun project, but also a challenging one, in that you have to bring the right people onto your team to be able to accomplish what you're trying to do. Thank you for your attention.